so to continue our discussion of DNA markers, it's time to look at how we analyze and what applications we have for DNA markers. All right, one of our analysis techniques is what we refer to as our coalescent model. In my opinion, the text describes the coalescent model in an overly complicated fashion. But essentially, the coalescent model simulates a most likely or probabilistic lineage pattern that describes the current observable variation. Ultimately, it links all observed variation to a shared common ancestor. And the probability of coalescence decreases with increasing population size. So in other words, as the population size increases, it is less likely that all observed variation will coalesce into a single common ancestor within the generational parameters. Now we have what are called our mismatch distributions. So this is the incorporation of mutation predictions into a coalescent model. So there is a direct comparison of individuals' DNA to one another. And a mismatch is when one individual has a mutation and another individual does not have the mutation. And overall, it estimates the amount of heterozygosity in the population. And this procedure is done using the FST to calculate the fixation index. So it's important to be able to interpret mismatch distributions. So if you look on the y-axis of this mismatch distribution, it is representing the frequency at which the number of differences occur. And then along the x-axis is the number of differences in haplotypes. So if we look at two haplotype differences have the highest frequency in our distribution in the population. The dotted line then represents our expected from our model, and the observed is the darker line, which shows what we actually found in the population. All right, so there are a lot of demographic effects on our mismatch models. Population size has a dramatic effect on the shape and appearance of the genetic mismatch models. Particularly problematic is when investigating past populations where population contractions or rapid expansions are temporally unknown. Furthermore, natural selection can produce similar patterns that cannot be differentiated from population size effects. So we need to then discuss what are the utility of using these methods. So overall, it's a powerful tool for the prediction of genetic relationships when investigating DNA markers. However, there are some weaknesses. Many variations on these methods of analysis are not directly comparable. So what this means is that differences between assuming a static population size versus one that used a dynamic population size creates widely different results, meaning that studies that use one method cannot be compared to studies that use the other. All right, so now it's time to move into the application of DNA markers in human variation studies. And our first stop is going to be looking at peopling of the Americas. Now, early models were based on linguistic evidence and dental traits, and they were secondarily supported by our blood group polymorphisms. Now, contemporary DNA marker studies have focused on the RFLPs located in the mitochondrial DNA. And they've identified 5 haplogroups, A, B, C, D, and X. These haplogroups are defined by a specific combination or presence of a series of mutations. So for example, haplotype A is defined by the presence of HAE3 restriction site. So note that haplotype A individuals may be remarkably different from one another at other points in the DNA, as only this point is used to define haplotype A. All right, there has been a lot of controversy over the X haplotype. And the reason for this is it has been used to argue a link between West Eurasia, so Europe, and the Americas, as it provides a possible explanation for European-like skeletons of some Paleo-Indians. However, the early Europe-American connection hypothesis has been rejected for the time being, and it's argued that drift explains the absence of the X haplotype in East Asia and its presence in Europe and the Americas. Now, there are some shortcomings for using only mtDNA. The reason for this is that mtDNA provides a limited view of human genetic history, as it only provides a window on a single locus, and it only follows the maternal lineage. Now, Y chromosome studies are equally as restricted as mtDNA investigations, and the reason for this is they only follow a patrilineal lineage. However, mtDNA and Y chromosome studies that use both are more robust in explanatory power, as they explore both maternal and paternal lineages simultaneously, and it's particularly powerful at exploring postmarital residential patterns. Now, future studies are beginning to look at autosomal loci, which are more difficult to study but have a more nuanced understanding of population genetic history. So now it's time to look at the application of haplotypes to human variation studies. The example we're going to talk about is the Cohen modal haplotype. So this was defined by the presence of an ALU insertion polymorphism as well as a microsatellite known as DYS19. And it's expected to be present in Jewish priests whose lineage is traced back to the original priest. The modal haplotype was identified in Israeli, Palestinian Arabs, Northern Welsh, South African Bantu speakers, and North Cohen Jews. So there are several interpretations over the Cohen modal type. So the haplotype is not specific to the priest lineage in Judaism, and it is reflective of a broader Semitic, so people associated with the Hebrew and Arabic languages groups. To some degree, it reflects population movements of these groups throughout the region, the exception being the Welsh, which is still a large unknown. Now, Judaism is not expected to have specific genetic markers, as religion is not genetically inherited, and Judaism, like many religions, remains open to conversion, which would naturally increase the genetic variation of the Jewish cultural group. All right, so there are some complications with using DNA marker studies as a means to exploring human variation. First, there's a vast amount of variation in how you actually analyze DNA, which makes it difficult, if not impossible, to compare across studies. And as such, it is extremely hard to create a synthetic argument or definitive conclusions that define patterns of variation while looking at DNA.